They do us a lot of harm. Now, these numbers on worldwide deaths are somewhat out of date. This is data going back from around 2003, 2004, and a lot of things have changed since then. I'm trying to get a more up-to-date table together for you, but it's a little hard to do. But this is showing, as of the early 2000s, the number of worldwide deaths that were attributed to these different types of disease organisms. When you hear about acute respiratory infections, we have a more common name for that. It's pneumonia. And there's a lot of ways to get infections in your lungs, deep in your lungs, that can kill a person. Collectively, back then, they were killing around 4 million people a year. And then you had HIV all by itself, killing a huge number of people, mostly in southern Africa. Of course, it's worldwide, but that's where it's really centered. Different kinds of diarrheas, such as amoebic dysentery or cholera, would be standout examples of deadly diarrheas. Tuberculosis, malaria, measles, killing a lot of people. And of course, we're getting some resurgence of that because people aren't getting vaccinated. Certain other things, like other childhood diseases, like whooping cough. Again, we have vaccinations. Everyone should get that. Tetanus, even syphilis showing up on this list. I think of that as kind of an old-time disease, but it also kills quite a few people. But again, these numbers are somewhat out of date. Regarding AIDS, the situation is quite a bit better now. We have gotten effective antiviral drugs that do a lot to suppress the harm of AIDS. And so you have, in places including in southern Africa, you're getting a lot of people that are getting these anti-AIDS drugs. And that has slowed this down a lot. Instead of 2.7 million, it's about 1 million deaths a year. And then with tuberculosis, malaria, they're also about 1 million. So the big three worldwide now would be AIDS, TB, and malaria. But generally, the numbers for infectious disease are better than we saw on this list. They're better now. And instead, more people are dying from diseases of old age, like cancers, for instance. So I'm just putting that in as a caveat here that these numbers are out of date, and in some ways, things are getting better. But given this list, what types of organisms appear to be the worst? Bacteria? Well, they show up in more of the lists, and so bacteria would be obviously something to focus on if you were trying to knock out disease organisms. Viruses may be in a close second place. Protists, we're talking about eukaryotic cells, such as paramecium, some of the other things that you looked at. Certain of those are harmful. Notoriously, you end up having malaria caused by one of those. So that's a eukaryotic parasite that can get into our cells and destroy our red blood cells. But mostly when you're talking about infectious disease, the main things we worry about are bacteria and viruses, and with good reason. So let's take a look at some things, starting with viruses. What is a virus? What is that? Are they like really tiny bacteria, prokaryotes? Are they more elaborate, or neither one? Yeah, they are neither one. They are not cellular. Go back to this. These are different descriptions of how to make a cell. And viruses aren't made of cells. They are made of some of the pieces that go into cells, but they're not things that have a cellular structure. And with that, they don't do most of the things that we associate with cells. For instance, they're not doing energy-releasing reactions like respiration. They are not doing chemical transformations that we would do in our metabolism. They're not taking in resources, rearranging molecules, expelling waste. They don't do any of that. They don't grow. They don't produce offspring. So most of the things that we think about that a life form's doing, they don't do them. So if you took a list of 10 typical characteristics of life forms, maybe they do 2 or 3 of those, but that's it. So I think of them as being non-living. Some other biologists will say, well, they are, in a very narrow way of looking at it, maybe they're sort of alive. Others will say not. I say that they're not alive, but they're made out of some of the stuff that life forms are made out of. So they're right on this kind of threshold, gray area between living and clearly non-living. What they are instead, they're the ultimate parasites. They are able to invade a living cell, and instead of making their own viruses, the way most life forms make copies of themselves, viruses take over living cells and force them to become virus factories. So living cells do all of this metabolic work for them to go and actually make copies of the viruses. That often leads to the death of the living cells. The viruses will often have these odd geometric shapes that don't even really look like life forms. Cylinders, geometric figures like that. So what you get in every case with a virus is there is some sort of a protein capsule that contains the genetic material of the virus. That genetic material can be either DNA or RNA, and some variations that are different than we're used to seeing with those. And sometimes there is an outer membrane, a lipid envelope that surrounds the virus, and that makes it, I suppose, look a little bit like a cell, but they're not using that lipid envelope the way a cell does. A cell is using it partly to regulate entry and exit of wastes and nutrients. They're using this basically just as a way to penetrate a living cell, because if you have a living cell that's already surrounded by a lipid envelope, it's pretty easy to get another one to bind to it and fuse with it and release the DNA into the cell. So that lipid envelope is mostly a way of penetrating the next victim. Here's one type of virus that's been well studied. It's a plant virus, one that infects tobacco plants. So you have this capsid that's a cylinder made out of many repeating protein molecules bonded together. And this one is an RNA virus. It has an RNA molecule inside, which it can release into tobacco cells. Here's a different one. This is one that infects us, adenovirus. And it has this regular geometric shape where you then have the DNA, in this case, inside of a protein capsule. And it has these protein spikes. These are specialized to allow it to bind to certain cells that it will then penetrate. So viruses will typically have proteins on the surface that allow them to lock on to particular kinds of cells as a way of trying to get into them. This one would get into things like lung cells. And then this one is a different one, herpes virus. That's an enveloped virus. And so that includes the capsid, DNA for the virus, and also the lipid envelope with more of these protein spikes for, again, binding to cells. In the case of herpes, that could be, say, cells, for instance, in the mucous membranes of your mouth, or maybe on genitals. So there are many of these viruses, and we classify them kind of like we do with living things into families and genera and so on. So there are international committees that govern this sort of thing, and they'll classify them as to having different sizes, different structures. The type of nucleic acid molecule they use, is it DNA, is it RNA, and some variations on that. How it is that they actually are reproducing inside the host cells, what hosts they will end up infecting, how they go through their whole cycle of infection. For instance, some viruses destroy the host cell and others don't. There are thousands of different species of viruses as they've been defined. And we can see what some of those are here. Often, they'll have a fairly narrow range of hosts that they're able to infect, maybe just infecting certain kinds of cells. You get something like HIV. Would that probably infect any human cell it comes across? What does HIV destroy in us usually? Yeah, it's destroying certain of the white cells or the immune cells. So it's something that favors just certain types of cells in humans. In that case, cells that are vital to our immune defense. So HIV is very specific. Some are able to go between species, and that's the thing we get concerned about when we look at possible new versions of the flu that might cause a pandemic and possibly kill a large number of people, jumping from various animals that humans spend a lot of time with, like with people that are, let's say, farmers that have chickens that they're around a lot. Sometimes there are avian flus which could make it into people, or other flus that can come from pigs and get into people. These are things we definitely get concerned about with future deadly pandemic viruses. So some viruses do get from one species to another. There are a lot of different families of viruses, and that's what I'm going to take you through is a list of these. Many of these cause famous diseases in us, like smallpox, AIDS. And then there are other viruses attacking all other life forms. There are ones that specialize on plants, many that attack bacteria, and we're going to see some examples of those. But here's some of the viral families that are affecting animals. Now, here's the type of nucleic acid. This is indicating whether or not they have the lipid envelope. Some don't have the envelope. In this case, here's one that infects us, causing a number of problems. And here we've got double-stranded DNA. That's like DNA that we're used to. Single-stranded RNA, again, like we're used to seeing. But there are some variations. This one, filoviruses, cause one of the most famous diseases, Ebola, fever that shows up in West Africa. In that case, that's an extremely deadly infection, and that's an RNA virus. So what the virus is actually putting into the human cells is an RNA molecule, but then it's able to do massive damage. Other types here, looking for one of the odd ones, right there. That's a weird one. Paraviruses, causing some diseases in humans and dogs. Single-stranded DNA. That's different from DNA that we're used to seeing. This is looking kind of like a messenger RNA molecule, but it's made out of DNA instead. So that's different from what shows up in living cells. Living cells have double-stranded DNA. That one is different. There are also some viruses that have double-stranded RNA. That, again, is different from what's normally in our cells. So viruses have this whole weird mix of different kinds of genetic information. And this one, historically, that is one of the most important ones. Smallpox. That is probably the biggest disease killer of humans of all time. That probably has killed more people through history than the Black Plague did. And we wiped it out. It's gone. That didn't happen by accident. That was a coordinated effort involving things like the United Nations, and they managed to track it down and vaccinate enough people in the affected areas that eventually smallpox had nowhere else to go. Smallpox was a horrible killer of people, but it only infected people. That was its Achilles heel. If we got enough people vaccinated against it, it had nowhere else to go. A lot of these viruses are able to live in other species. With Ebola virus, we know it's normally in other species, and we haven't quite figured out what those species are. But Ebola is something that doesn't linger in humans. If it gets into humans, you either die from it or you recover. It doesn't just hang around in some sort of quiet latent form. So most of the time, no humans are infected with Ebola. And that means it's gotta be in some other species when it's not in us. It can go back and forth. Smallpox couldn't do that. And we vaccinated enough people in the right places that it finally was extinct in nature in 1977. That was when we had the last case of that occurring. Since then, we have samples of it in research labs in places like Moscow, in Maryland, and unknown if it's anywhere else in the world. But there is certainly a fear that it could come back if somebody decided to use it as a weapon. It was never wiped out as far as the research samples, but it's not out in nature and no one's getting infected with it now. They estimated in, say, the 1800s and the earlier 1900s, maybe 500 million people worldwide died from that, and it was a horrible way to die. And after 1977, no one has. So that is probably the biggest triumph of public health of all time, that eradication. When they talk about it, they write eradication with a capital E. It deserves that. That was a one of a kind effort that's not been repeated with any other disease. But when it came to doing the eradication, they were smart. They took on the biggest enemy that we had, and the public health people won. That took an enormous effort by a whole lot of dedicated people taking a lot of risks. And it was really heroic for them to do. As a result, you guys basically don't have to worry about getting this horrible disease. I was vaccinated against it. Has anyone here been in the military? No. If you had been, you would have been vaccinated against that and also anthrax. Those are two that the military is concerned might be used as biological weapons. But otherwise, people generally don't get vaccinated against it. Let's just hope it never comes back on the scene. But that disease, luckily, is gone. HIV, well, that's causing AIDS. That is another, I believe, RNA virus. It's what we call a retrovirus, and we'll see those in a bit. There's also viruses affecting plants. Here's some examples of those. Here's another one of the weird ones, single-stranded DNA. And I'm pretty sure there are some viruses that are double-stranded RNA. So anyway, many, many different viruses out there. Here's an example of one. That's what's called a bacteriophage. This is a virus that infects bacteria. When you hear this term bacteriophage, don't think that it's a bacteria. It's not. It's a virus that infects bacteria and destroys them. So what you have is this structure, this capsid, that is protein containing viral DNA, and then we have these structures including the tail fibers. This is able to attach to a bacterium, and then this works like a syringe, injecting the DNA into the bacterium. That DNA will then try to take over the bacterium. Here's a bunch of them doing it. Here is a living bacterial cell. This is the bacterium. It's huge compared to them. You can't see these with light microscopes. But there we've got the capsid, we've got the tail fibers, and here's the DNA being injected into the bacterium. These three are doing it, so is that one. Here, empty capsid is already floating away, having done its job. The bacterium may be fighting back. Remember those restriction enzymes we used in the lab? Bacteria have those to defend against this kind of thing. Their own DNA is protected. And then foreign DNA coming in, they can try to destroy it if they choose to. They may or may not, but they can try to do that. And so sometimes the bacterium will win the battle, but this one's probably being overpowered. And certain of these bacteriophages destroy the host cell, completely destroy it. That's known as the lytic cycle. Lysis. Lysis is rupturing, destruction of a cell. That's what this does. And we get these ones that are known as T-even bacteriophages. Ones of these types, T2, 4, and 6. These are bacteriophages that invade the bacteria and kill them. You have a single molecule of DNA, normal DNA in there. The tail has its recognition proteins able to bind to the E. coli cell. And what it will do then is, if the DNA takes over the bacterium, if the DNA wins, the cell will go into the Leydig cycle. What it will do, it'll have the viral DNA inside the cell, and those genes take over the functioning of the cell, causing the cell to start making copies of the virus. Part of what will go on is that the cell will digest itself in the process. The cell, the bacterial cell, has its own DNA, but that's made out of nucleotides, which could be used to make viral DNA instead. And so the DNA will actually, of the cell, will get digested and turned into viral DNA. So the cell is just being hollowed out, losing its own identity. And then finally the viruses are assembled inside the cell, and it ruptures, and so it explodes and releases more viruses. Then they can go and repeat the process. So there's a reason that people will sometimes talk about the zombie virus or the vampire virus. It's a good analogy to what actual viruses do. As far as a victim getting bitten and then going on and infecting others, that's basically what's going on here. So you have one of these really virulent viruses attaches to the bacterium. There's the bacterial DNA. Viral DNA gets in, commands the digestion of the cell and production of new copies of the virus. We end up making many copies of the viral DNA. We build the other parts of the virus out of proteins. The cell is hollowed out, turned into a container that has maybe 200 viruses in it, and then the cell wall digests away, ruptures, releases the viruses. That's the Leydig cycle. Summarized here, same idea. That's not the only way that things can go. You can also have a quieter version called the lysogenic cycle, and that is where the bacterial or the viral DNA gets into the cell and it doesn't attack the cell. Instead, the new DNA gets incorporated into the DNA that's already there. So you have now genes from the virus being inserted into the bacterial chromosome that was already there. And then, when this cell divides, it's copying everything. You're making copies of the viral DNA. The virus is able to spread in this way, and it might stay that way for a while. This has a disadvantage. This is not allowing it to go and infect new cells. So all you're getting is copying of the DNA as you're producing new cells. And we will have what are called temperate viruses, and they will actually go back and forth between these two cycles. Here again is the Leydig destructive cycle. Here's the lysogenic cycle. What can happen is DNA from the virus gets into the bacterium, and initially it's incorporated inside the bacterial cell as what we call a prophage, prephage, something that could become the bacteriophage, but right now is not. It's being copied here. You're producing copies of it under the right environmental conditions. This prophage can emerge and become active and take the cell into the Leydig cycle. So if there is some sort of environmental trigger, we end up emerging from this quiet, latent stage and going back into the aggressive stage that leads to destruction of the cells. Now we get some diseases in us that do this. This is a bacterial model, but we get this kind of thing happening in some cases with human diseases because we get some cases where the viral DNA gets into us and enters our chromosomes, and we're never rid of it. It stays in us for our lifetimes. And from time to time, you may get an outbreak where the disease becomes more obvious. Do any things come to mind that might fit that model? Some sort of disease that would periodically flare up and other times hide? Oh, with cold sores. Yeah, cold sores, herpes virus. If somebody is infected with herpes, let's say they've got the oral kind, so they kissed somebody who had a cold sore or the remnants of one, and they picked up herpes viruses, and the herpes virus infected some of the cells, basically getting into the nerves near their lips. Well, they might have had a big flare-up of this initially, and then the disease went away. And it seemed like, okay, well, I had that one flare-up, and it's been years, nothing has happened, it must be gone. But it's still there in their cells, hiding out in the nerve cells, nerve ganglia, near the lips. And under environmental stresses, that DNA can reemerge and lead to explosions of cells, which is what you're getting with, say, a cold sore. So with a cold sore, what could be the environmental trigger that causes the DNA to reemerge and become active with a cold sore? Cold. That'll happen if, say, a person is getting another infection and their immune system is all tied up with fighting a cold or fighting a flu. And if they already were infected with the herpes virus that was lying latent in their cells, it can reemerge, basically thinking, now's my chance. The immune system is busy fighting this flu virus. Now the herpes virus can run amok and not worry about being destroyed by the immune system, not as quickly anyway. So under sort of environmental stress, your cells around your mouth, for instance, can switch from latency to having an active infection leading to a sore, which gives it a greater chance of getting into another victim. Now, in general, these viral particles will, when they're leaving our cells, most commonly, the viruses that infect us are enveloped. And so if a cell creates a viral particle, it passes through the plasma membrane and picks up a piece of it. And so you'll have the capsid, which contains the viral DNA. It's surrounded then by a piece of membrane along with recognition proteins to bind to the next cell. In this case, we're getting some of those produced. These are flu viruses. These will have recognition proteins that are able to bind to the receptors that we have for a neurotransmitter. We have a neurotransmitter called acetylcholine. That's where nerves can talk to muscle cells. Well, that receptor that's on many of our cells is something the flu viruses are able to attach to. So our cells are not making those receptors with the intention of being infected. This is just something the virus takes advantage of. There's a lot of different ways that that viruses can cause harm. Production of enormous numbers of the viruses can destroy the cell, as we were seeing with the bacteria. Also, when cells are breaking down, when they are dying from this sort of infection, that can then cause inflammation, fever throughout the whole system. And sometimes viruses will actually alter the genes. If they are moving into your genome, they may actually go right in the middle of a gene and possibly disrupting its function, potentially leading to cancer, for instance. Well, here's one infection, one of those childhood diseases that we vaccinate against, mumps. That's an RNA virus. So you have a lipid envelope around the whole virus, and then it has a protein capsid, and inside of that, an RNA molecule. That penetrates the cell. Once this capsid with the RNA has gone into our cell, the protein breaks down, releases the viral RNA, and then given that it already has an RNA molecule inside our cell with the viral genes on it, it starts making copies. There's a viral enzyme that comes packaged in the virus that tells your cell to make more RNA copies from the one it already has. We don't usually take one RNA molecule and make more RNA from that. But these viruses bring in the enzyme that our cells then start using to make copies of the viral RNA. The copies of the viral RNA are then used in turn to make more copies of viral proteins. So you end up getting more copies of the viral proteins, more copies of the viral RNA, and ultimately assemble those together to make more viruses. You build viruses using these proteins that were made according to these instructions, and then package in more copies of those same instructions. They leave the cell, stealing a piece of plasma membrane on their way out, and so you go from having a virus penetrating the cell, releasing viral RNA inside, making copies of it, making proteins, doing translation from this viral RNA, making copies of the viral RNA, building new viruses, and out they go. That's an infection that can cause deafness in children, so it's an important one to vaccinate against. Now, we are not able to kill viruses with antibiotics. Antibiotics are poisons. They attack living processes. Viruses don't have any living processes to be poisoned, to be gummed up by an antibiotic. You give them something like cyanide, they don't care. Cyanide interrupts cellular respiration. They don't do respiration, and so antibiotics won't touch them. So antibiotics are often good against bacteria, but they're useless against viruses. So with most viruses, we just have to treat the symptoms. So we'll go and give somebody care to make them more comfortable and deal medically with more important issues, like if they have a raging fever, you can do things to try to reduce that. But as far as destroying the viruses, that mostly has to be done by their immune system. We have developed some specialized antiviral drugs, starting with herpes. That was about the first one where we got one of these. And so we have successful antiviral drugs that interfere with viral replication. These are things that are basically drugs that are able to prevent the virus from making more copies of themselves. So we'll be doing things like, for instance, interrupting the replication or certain other aspects of the viral life cycle. So this is not acting to destroy the viruses, but it prevents them from making copies and doing more harm. It gives your immune system more of a chance to actually deal with the infection. Retroviruses. That's where we get HIV. And this is, of course, a noteworthy one of the top killers worldwide. In that case, it's enveloped. It has RNA genes inside. It gets into our cells and produces a DNA molecule. If we're doing normal transcription in our cells, we're going from what to what? Transcription starts with something and ends with something. DNA to RNA. Here, we're going from RNA to DNA. It's backwards, retro. That's where the retrovirus name comes from. So what we have, our cells normally can't do that. They normally couldn't take an RNA molecule and make a DNA molecule, but the retroviruses come with an enzyme that will do the job, reverse transcriptase. It carries out transcription in reverse. Given an RNA molecule, it will build a DNA molecule, and that DNA gets inserted into our chromosomes. So what happens is HIV infects a cell. Then the cell will take that single-stranded copy of the RNA, use reverse transcriptase to build a full-blown DNA molecule, and inserts that into one of our chromosomes. This is part of the process of ultimately breaking down our immune cells, and I wish I knew more of just how that happens. I don't. But that DNA gets into our cells, and it's kind of like the prophage with the bacteria, a predecessor of an actual phage. In this case, it's a predecessor of an actual virus, because the cell is now capable of producing more of the viruses once the DNA is inside of it. So starting with this is the big enemy. That's HIV. You have this capsid, a little fatter at one end than it is at the other, made out of viral proteins. 2 copies of the virus's RNA, also 2 copies of the reverse transcriptase. And it goes inside of a lipid envelope with these recognition proteins on it that can bind to immune cells, like the T cells in your immune system. That will infect one of the T cells, and you'll get usually some initial symptoms which then subside. So a person may have been infected and then feel like, oh, I have a cold, and that goes away after a while. Person can be healthy for years, but this spreads one to another of the immune system cells and gradually knocks them out, ultimately destroying the body's immune system and leaving you vulnerable to other infections. So people are not killed directly by HIV, they're killed by other things like pneumonias that can then invade them once the immune system has been wrecked. So HIV infects the cells. There's the viral RNA. Reverse transcriptase goes to work until we now have double-stranded DNA. That gets inserted into a chromosome, and from time to time, it's activated and produces more copies of the virus. The virus will leave one immune cell and spread into another, or maybe infect another person. And here's one of them budding off, leaving one of our cells. So you've got here an immune cell, and there goes that bad boy on its way out. That's another HIV particle. So that is a lot of what goes on with viruses. I think I'm going to leave this part right there. We'll look at bacteria on Monday, and I'm going to give you some questions to work on.